Hi everybody. The focus of our lecture today will be on cell division, more specifically a type of cell division known as mitosis. So if you take a look at the cells you have in your body, we break down the cells to basically cells known as somatic cells and cells that are referred to as reproductive cells. Somatic cells are basically any cells you have in your bodies except sex cells. So hair cells, skin cells, eye, heart, etc., etc. So to divide these cells, we use a technique or a process of cell division referred to as mitosis. However, when we talk about sex cells, utilized for the process of reproduction, we rely on a cell division process referred to as meiosis. Now, our focus for today's lecture would be the division of somatic cells using mitosis. Now, a couple of reasons we undergo mitotic process to create new cells are growth and repair, as well as replacing dead cells. So basically the idea behind cell division is you start with one cell and at the end you create two identical daughter cells. If you take a look at the picture I have on the side, you notice that the process of cell division is divided to two main processes. One that is represented in about 3/4 of the circle, and that is referred to as interphase, and itself has three parts. And then you have about a quarter of the process of cell division is a process referred to as mitotic phase or M phase, which is also broken down to smaller fragments. So we are going to start talking about interphase and then work our way through the M phase or mitotic phase. But before we discuss that, what I want to mention to you guys is a couple points about the genetic information, which should be more of a reminder to you from our previous lecture. So, starting with the genetic information that is found inside your cell, the type of genetic information we had was chromatin, which is basically DNA plus protein. We have chromatid, which are basically condensed DNA. Oops, sorry, DNA, not DNA. And then chromosome, which is basically two chromatids. Now, when we look at a cell that is not undergoing cell division, not planning to undergo cell division, genetic information is found in the form of chromatin. If DNA starts to condense and it requires to undergo the process of cell division, DNA is condenses to form a structure called chromosome, which are basically two sister chromatids. So let's walk through this and hopefully this makes sense as we go through them. The process of interphase, the first process in the cell cycle, is a process referred to as interphase. Interphase is where you prepare the cell for becoming two daughter cells. It is made up of three steps: gap 1, which is shorthand to G1, synthesis, S, and gap 2, which is shorthanded to G2. Now, during gap 1, cell is undergoing increase in size. It produces new proteins, new genetic information, new mRNA, and it's getting ready to duplicate its genetic information so it can spread it between two cells. So basically, gap 1 is a step that is preparing for the process of DNA duplication. During S phase or synthesis, your DNA is being replicated, and during gap 2, you basically continue to grow cell, and you also have checkpoints to make sure that the genetic information has been duplicated properly. Because if these genetic informations are not duplicated properly, it's going to cause damage to the cell, cause abnormalities that could possibly lead to disease such as cancer. So simply put, during interphase, you grow the cell, you make all of the extra components that you need, you duplicate the genetic information, you make sure the genetic information is okay, and you again continue to grow the cell. As you notice, the genetic information during this stage is still represented as chromatin, and the nuclear envelope surrounding the nucleus is still present. As we start to undergo the process of mitosis, we start with the first process, which is referred to as prophase. That is followed by a process known as metaphase, anaphase, eventually telophase. And when telophase is done, and as telophase is finishing up, you start a process to divide those cells to two completely separate cells through a process called cytokinesis, where your cytoplasm is also divided. So the goal during the process of mitosis is to separate the genetic information evenly between the two daughter cells. So during prophase, which is the first step of mitosis, the following takes place. Your chromatin starts to condense to form chromosome. So during the chromatin, if you look at it under the regular microscope, you will not have any visible DNA. Rather than what you will see is basically a background shading which doesn't identify individual genetic information or individual components of the genetic information. However, when genetic information condenses to chromosomes, it becomes visible under the light microscope. As you can see, this is basically genetic information start to form chromosomes, and this is by the later stages of prophase, the chromosomes are very visible as these long stringy structures. Something that it needs to be clear, again, by this stage, genetic information has been duplicated, so every chromosome contains two chromatids, two sister chromatids, which means that they have two sets of genetic information. The duplicated genetic information from last time is present during this phase. So chromatin is uncondensed genetic information. It gets duplicated during interphase, and when that duplicated genetic information condenses, it forms two sister chromatids in the form of a chromosome. Notice that if you want to spread the genetic information between the two cells, you need to remove nuclear envelope. So as you can see, nuclear envelope start to dissipate. And lastly, the two centromeres that are responsible for formation of a structure called spindles, they usually are present on one pole, and they start to move in opposite poles and start to form your spindles that are essential for separation of the genetic information. Spindles are basically going to be microtubules that are being formed from your centromeres. Here again is the highlighted concept of chromosome, chromatin. So if your genetic information is uncondensed, this is like your typical DNA, double helix. You start to wrap it with these circles, which are your proteins. It becomes chromatin. Chromatin can be condensed to form arms of the genetic information called chromatids. And two chromatids together will form your chromosome. And they're connected in this central space called centromere. Now, if we continue, the next step of mitosis is a step called metaphase. So notice that the spindles that you see is this yellow line has connected to the centromeres, and through their movement of the spindles, they have placed the genetic information you can see in green and purple at the central line of your cell, which we refer to this as metaphase plate. So during metaphase, the genetic information basically line up at the center of the cell. You can see this has these blue strings at the center. Again, the green in this picture represents your spindles that holds their chromosomes. At this stage, because you want to separate the identical genetic informations from each other, you need to basically start to chew through the centromere. And therefore, what you will have is you have enzymatic activity that starts to separate those two sister chromatids. During the next process of mitosis, your spindles have connected effectively to your centromere. Well, they are already actually effectively connected to the centromeres, but the two sister chromatids are separated, and the spindles start to pull them in the opposite poles. So you see this first half of the chromosomes are being pulled to the top half, and this half of the chromosomes are being pulled to the lower half of the cell, or the opposite pole. Notice, because centromeres are literally pulling the genetic information or the chromosomes right at the center, you create those V-shaped patterns. The example that they usually help you understand this is, think about that you have a handkerchief or a scarf. You hold in the center of your scarf and you hold your hand outside a moving car, and you can see that the location where you're holding is basically is the connection for the spindle, and the two ends of the scarf which form that V shape is basically the same pattern that's created for the chromosomes as they're being pulled by the spindles. Now, the other thing that is happening is you can see some of these spindles are starting to expand farther and farther, which are eventually going to basically pull the cell in farther and farther apart, and it starts the process of separation to form two separate daughter cells. So this is basically, as the spindles are starting to be basically pulling the cells apart, these start to create the separation between two cells. So in this picture, you have one visible cell, and in this picture, you can see that the cells have been elongated and they're starting to form two separate cells. Now, during the stage known as telophase, this is basically, you have separated your genetic information to two cells, and now you want to return your cells to their original state, which means that the chromosome needs to be uncondensed to form chromatins, which is what happens. So you go again from a visible genetic information to an invisible, quote-unquote, chromatin. Your nuclear envelopes are going to be formed around your genetic information. And since the job of your spindles have been finished, spindle starts to break down to their original tubulin subunits. Now, as this process of the telophase is coming to an end, cytoplasm of the cell also starts to separate by forming this furrow that you see at the center, right here and right here. Basically, this is referred to as a cleavage line or cleavage furrow. And it starts actually, as I mentioned, starts throughout the anaphase as the spindles start to be basically pulling the cell and elongating the cells. And it continues throughout the telophase until you separate your cytoplasm completely and forming two identical daughter cells at the end. Here's basically the steps in one picture. You have your interphase. Notice that the genetic information is basically look like a long string that is tangled. These are your chromatins. You have your centrosomes that contains and eventually becomes your spindles. During the next phase, which is your prophase, you start to condense your genetic information to your chromosomes with the sister chromatids connected using centromere, and nuclear envelope is starting to dissipate and your spindles are starting to form. During the next stage, which is metaphase, this is pre-metaphase or pro-metaphase, and this is metaphase, genetic information line up at metaphase plate. Using the spindles, you pull the genetic information in opposite poles during the process of anaphase. And lastly, during telophase, your process of prophase is reversed, which means chromosomes are starting to become chromotins, your nuclear envelope is starting to form, and your spindles have disappeared. At this stage, your cell is also start to divide as early as anaphase, and throughout the telophase, you start to separate your cell. During the last stage, known as cytokinesis, your cleavage furrow is completed, and your cell is divided to two identical daughter cells. Note that the genetic information in these two daughter cells is identical as a result of duplication that happens during interphase. A couple of points. As I mentioned, the genetic information is identical to the original cells. This process is used to repair and grow, as well as replace the dead cells. If you have out of control divisions of cell through the process of somatic cells, I should say, you basically have some sort of cancer depending on the tissue. And interestingly or sadly, adult nervous system cells cannot undergo this process, and therefore damages that happens to your nervous system and your neurons will be on a permanent base. And that has to do with a structure called growth factors. I will not discuss the meiosis with you. I do want you guys to know a few points about them, but nothing in detail. Understand that meiosis is associated with your sex cells responsible for production of a sperm and egg. It has two stages, meiosis 1, meiosis 2. Instead of forming two identical daughter cells, you actually form 4 cells that are not identical, but only carry half of the genetic information as the original cell. Genetic information are not identical because you undergo something called crossing over, where the genes are being basically shuffled from one chromosome to another to create diversity. And that's the reason why you won't have two kids, even though their siblings and from the same parents, they do not look identical to each other. One quick point that I need to make about this, and again, you guys are more than welcome to review these to know more about them, is the following. When I'm talking about the process of, when I'm talking about the process of mitosis, this is the actual genetic information. So in a cell that you start with, you have 46 chromosomes. 23 comes from your mom, and 23 comes from your dad. During the interphase, you change the 46 chromosome to 92 chromosomes because you're undergoing basically genetic information duplication. And at the end, you take that 92 and you spread them again between two identical cells. Each of them have 46 chromosomes. Now, in the process of meiosis, you still start with a 46 chromosomes, which are again 23 from your mom, 23 from your dad. It does duplicate to become 92. But the difference is because you have meiosis 1 and meiosis 2, you actually end up with, and this is a really short version actually, you end up with 4 cells, each of them having 23 chromosomes. You only have one stage of interphase, so genetic information will only be duplicated during meiosis 1. But by the end of meiosis 1 and meiosis 2, so this is 1 and 2, when you're done with both of the meiosis stage, you will end up with 23 chromosomes on inside either the egg and the sperm, which is a good thing because if you assume that, let's say, this is a sperm, the tail is the sperm, and it meets with an egg that is also have a 23 chromosomes, what you end up with is a cell, a fertilized cell, basically reproduction, that will have 46 chromosomes, which is the magic number we want for each of our cells. But if your sex cells have 46 chromosomes, then your cells will have double of the amount of genetic information when you had an egg fertilized by an sperm. So this is extremely important for you guys to know how many cells are produced at the end of each stage, or at the end of each of these processes, and how many chromosomes are present in each. And that's about it.